Okay, so today we have a final few thoughts about metabolism before we move into our next topic, thinking about cells and their membranes and the forces that make things cross membranes in and out of cells. Okay. So we've talked about how we have our typical aerobic cellular respiration, our normal metabolism, our normal way of producing ATP. Starting with glycolysis, getting us to ATP from out in the cytosol, out in the cytoplasm, moving our end product pyruvate into the mitochondria where we go through the Krebs cycle, making another 2 ATP plus a lot of reduced electron carriers. And then we enter the electron transport chain with the goal of doing oxidative phosphorylation, our third step. So in that process, we use electrons that we're pulling off of those electron carriers to power this movement where we push hydrogen ions into our intermembrane space of the mitochondria and let them rush back through like a waterfall, letting ATP synthase spin and make a lot of ATP. I'm going to ballpark it at like 30, because it's not very number. So that's a big bang for our buck there, but we know that we can produce ATP without oxidative phosphorylation. When we do that, what we do is we keep running glycolysis out in the cytoplasm over and over again, but to make sure that we still have enough NAD to turn into NADH, which still has to happen as part of glycolysis, regardless of whether we're actually going to use that NADH. We have to turn pyruvate into lactate. So that's our lactic acid fermentation, our kind of anaerobic way of producing energy in our cells. Anaerobic, no oxygen. We can do this in no oxygen conditions, which some of your cells will be in, even though you are breathing oxygen. Okay. So what we left off with was other things that we can break down other than glucose, because all of those were ways that we're processing glucose, ways we're oxidizing glucose, ways we're breaking apart the bonds in glucose to store energy in our ATP molecule. So we mentioned that we can do this with proteins. We're going to see that we can do this with fats. And here we have our special storage carbohydrate glycogen that we have throughout our body, but particularly important in the liver and kidney, because in the liver and kidney, we can take glucose that was stored in that glycogen and send it into our bloodstream, send it to other parts of the body. So we mentioned that there are a lot of words that look very similar unless you squint, right? So glycogen is our storage molecule, right? So this is a big branched storage molecule full of lots of glucose subunits. And so a bunch of glucose stuck together. So we can break down glycogen, we can lyse it. So that's our glycogenolysis. Okay, so if we're breaking down glycogen and we're trying to get glucose out of it, there are a couple intermediates would involve enzymes, right? Because first, we don't immediately get like good clean glucose with nothing stuck to it. We get glucose 6-phosphate when we pull those old glucose monomers out of our glycogen. And so we need an enzyme to turn this into normal glucose. So that's our glucose 6-phosphatase. Turns it into the normal glucose that can travel around your body easily and get into other cells. So this is a process that's in particular happening in the liver because we've got the right enzyme. Okay. Now we can similarly, right, make glycogen. Right? Sometimes you're eating a lot of carbs, you're eating a lot of sugars. You don't need that much ATP right now, so you're going to store those for later. So it's a similar process, right? So we take our glucose that we're eating. We have a different enzyme, in this case hexokinase, right? You see that ACE ending telling us that enzyme. It makes our glucose 6-phosphate, which then through the process of glycogenesis is gonna get us our glycogen. Right? So we can break it down, we can store it. We also had a slide talking about gluconeogenesis. So we can also make glucose from something that was never glucose. So we can take parts of other molecules and kind of process them a little bit until we build a new glucose molecule. We'll talk about this a bit more when we actually talk about our endocrine system. We're going to talk about insulin and its role in helping us do this by controlling our hormones when we're eating versus when we're fasting. So these words will come back later, which is why we're introducing them. Now, most of your energy that you're saving for later isn't actually stored in glycogen, because glycogen isn't that efficient at it, and we know that the glucose we have trapped in glycogen mostly can't get out of cells unless we're in the liver or kidney. Okay. So most of our energy is going to get stored in triglycerides, fats, because those are really effective. We can store a lot of energy in a triglyceride. So a reminder, our triglyceride is a type of lipid, right? So we have a glycerol backbone, and then we have our 3 fatty acids, right? We can have saturated fats, we can have unsaturated fats, we've got different types. We're not interested in those different types right now. We're just gonna have a generic triglyceride, whatever kind it happens to be. Now, if you were to weigh a triglyceride per weight, so for like the space it occupies, the density it is, lipids are going to have more energy stored in their bond than carbs or proteins do. They're really dense. They're a really good way to save this energy for later, but not have it take up too much room in your body. Most of these triglycerides are going to be in your fat cells, which are called adipocytes. Okay? So adipose tissue is fat tissue. These are the types of cells in that tissue. So now we got how it works. So when you're breaking down a fat, you're breaking down a triglyceride, you're going to start by peeling off those fatty acids. So that's why we're just drawing that structure. We've got our glycerol backbone. We're going to rip off those 3 fatty acid chains on it, and we're going to process those different components separately. So from our glycerol, we're going to convert that into this molecule, dihydroxyacetone phosphate. I will not ask you to say that. I don't know how many times I've said that aloud, but we're going to turn it into a molecule that can go into glycolysis, is what we do want to know. So glycerol can go into glycolysis. As it goes through glycolysis, it works a little differently than a normal glucose molecule would, but by the end of the process, as they go from glycolysis into the Krebs cycle into oxidative phosphorylation, you'll get about 20 ATP molecules per glycerol. The fatty acids work in a different way, and this is really where we're so effective at storing energy in the fat. So our fatty acids, those chains are pretty long, right? So the squiggles I was drawing, carbon on the points, and then we're just interpreting hydrogens as hanging off, right? So we got carbons everywhere. We got like a little zigzag, and we're just gonna assume that unless we say otherwise, there are 2 hydrogens hanging off, so the carbon has the 4 bonds that it needs to have, right? So the whole chain is like this. It's our hydrocarbon, that's our fatty acid chain. What we're going to see when we break them down through the process of beta oxidation, that's the name for this process of breaking down fatty acids. We're going to break it down like 2 carbons at a time, right? So we're going to take 1 carbon, 2 carbon, and then we're going to chop the molecule here. Right, we're going to break down 1 carbon, 2 carbon, chop the molecule there through the process of beta oxidation. And for every little 2 carbon chunk like that, we're going to end up getting 17 ATP. So 17 ATP for every 2 carbons, but those fatty acid chains are way longer than 2 carbons, right? So 17 is still less than 30 that we saw happening in our oxidative phosphorylation of glucose, but glucose, that's all you get from that molecule. For our fatty acid, honestly, I do not know how many carbons long that they are, but they weigh more than that, right? Big, they're bigger. Okay. So what's happening in our beta oxidation? We're chopping that fatty acid apart in little 2-carbon blocks. And each of those 2-carbon blocks is going to get converted into acetyl-CoA. Okay. Now we can do the rest of our process pretty normally. So that acetyl-CoA goes into our Krebs cycle. So we're going to be producing a lot of reduced electron carriers, right? We're producing a lot of NADH, we're producing some FADH2, right? And a little bit of ATP. All those reduced electron carriers, the NADH, the FADH, are going into our electron transport chain, which means we can do the oxidative phosphorylation step as well. So a little less effective per 2-carbon block when we're doing oxidative phosphorylation from a fatty acid, but because you'd be doing this so many times per molecule, your fat is going to get you an awful lot of energy. So fat metabolism is super important and uses some of these same cycles, some of these same processes, just jumps in at a different place, but we can chop a fat into a lot more pieces than we can chop a glucose molecule. But finally, we have protein metabolism. So mostly what we want to remember here is the name for this, proteolysis. This will come back later on when we talk about digestion and absorption eventually. All right, so proteo is for our protein, lysis is for breaking it down. All right, so we're pulling apart the amino acids in a protein. There are many amino acids in a protein. And then we're going to pull apart the amino acids themselves. Okay, so we pull apart our amino groups, we pull them off and leave us with a keto acid and ammonia. And then our keto acids can include like pyruvate or acetyl-CoA, which go into the Krebs cycle. So we get some energy from proteolysis as well. Not as effective as beta oxidation, but it can happen. If you're not eating carbs, you will still have energy in your body. The process is just a little less effective. Okay. So that's all we want to know about metabolism. And for those last couple of slides, we really just want to know the very basics. So there are a lot of word-specific intermediates on those slides. You don't need to know the intermediates, but you do need to know that we can break those down. Triglycerides are super effective. You want to know beta oxidation, and that we're getting a lot of energy for every little 2-carbon chunk. Okay. So we're going to take a second and think about plasma membranes now, because we're going to start thinking about what our cells are actually doing. We're going to be thinking about transport, and in transport, we're going to be thinking about one way that we use ATP, or we need ATP. We're going to think about active transports of using ATP to move molecules in directions that they don't necessarily want to go. They're going to start by listing the molecules that are in a plasma membrane. So I'll give you a minute to write down what you can remember about plasma membranes. We'll begin walking through it to check ourselves, beginning with our phospholipids, and then talking about some of the other pieces of the membrane and its behavior. So when we talk about the plasma membrane, the cell membrane, you may have heard this term, the fluid mosaic model. So the fluid mosaic model, meaning that our membranes are mosaic. So we have different pieces, right? We're imagining different tiles of different colors. This is because we've got phospholipids in there. We've got cholesterol, which is a type of lipid as well, steroid. We've got membrane proteins doing a number of different jobs, some involved in transport, some working as enzymes, some working as cell junctions. We've got a couple carbohydrates attached as well, helping us kind of signal to other cells, among a couple other jobs. Now it's fluid because it moves, right? So the mosaic part gets to the different pieces. The fluid part gets to this is not a rigid structure. It's not a cage. It's not a rock. It can move around. And that movement is part of how we see things moving across membranes and part of why cells can move the way they do. We're going to start at the beginning. So we've talked about phospholipids when we were talking about our different classes of macromolecules, right? So the phospholipids were the ones with a phosphate head, right? And those two fatty acid tails, right? So we had our phosphate head, it's polar, and it's hydrophilic, or water-loving. And our fatty acid tails are the opposite, right? So they're nonpolar, which means they're not attracted to water. They actually repel water. So they're hydrophobic. So like if you have a phobia, these fatty acid tails have a phobia of water. Okay. So the phospholipids are our basic structure for the membrane. They're definitely there, you definitely need them. They're the majority of the molecules you have. And they're lining up in this bilayer structure where we have a phosphate head, both outside and inside. And then we have this double layer of fatty acid tails as those hydrophobic regions kind of cluster together, trying to escape our water. Because we're remembering we have extracellular fluid outside the cell, our ECF, and we have intracellular fluid, our ICF, inside the cell, right? So there's water in both these locations. These phospholipids and their physical and chemical behavior is really important to how membranes work, because our phospholipids are really why many things can't get across a membrane without help, right? So we've got this thick, nonpolar hydrophobic layer that is going to prevent anything with a charge or anything polar from just diffusing through the membrane. It's going to be blocked, it's going to be repelled by this thick hydrophobic zone. And they are squished together, so anything too big, even though our membrane is fluid, can't make that big of a gap necessarily. But these phospholipids do kind of shift relative to each other. They can kind of jiggle a little bit. So this is part of what gives the membrane that fluid quality, that movable, malleable quality, which can be important if you're like an amoeba trying to eat something, right? Or as we'll see, if you're a neuron trying to release neurotransmitters, it's going to be important that you can change the shape of your membrane. Okay, so our phospholipids are impermeable to water because of that thick hydrophobic zone, right? So that piece in the middle where you got those fatty acids means water is not going to be able to cross, right? So we're still just talking about this part I circled before. So water can't cross this. However, unless I tell you otherwise, our imaginary cells are little circles that we draw. We're going to assume that they have a protein called an aquaporin in them, creating a passage for water. Okay, that's going to be important for when we think about like the process of osmosis, right? Most cells, effectively, water can cross because of this other type of molecule that's present in pretty much all of your cells with a couple exceptions in the kidneys. Okay, so we'll talk about exceptions to this in A&P2 when we get to the kidney. But for now, we're going to think about, phospholipids, and our kind of baseline for this membrane should be impermeable to water. If we think about stuff moving across, we're going to think about this as having kind of pores, right? Aquaporin, right? This is the pore part. It's a pore. For aqua, that's our water. All right. Then we've got our cholesterol. So this also adds a little bit to the fluidity of the membrane and kind of jiggliness, because basically it's a different shape. So our cholesterol is that little yellow bit in there, right? So this piece here, so we can recognize that basic steroid structure of those kind of four rings pressed against each other. So this prevents molecules of the same type, okay, the phospholipids, from kind of crystallizing, and it could get trapped, locked into exactly the right position if it was just them. The cholesterol breaks that up a little bit. Yeah. So it interferes a little bit between their reactions with their neighbors, making sure our membrane stays nice and pliable. There are also lipids. Lipids are broadly hydrophobic, right? So broadly repelling water, thinking oil and water when we're thinking lipids. So this cholesterol is also kind of blocking water from getting through the membrane, right? So decreasing the permeability of the membrane through water. So permeability means we're thinking about what can go through. If something's permeable to water, if our cell's permeable to water, we mean that water can travel through the membrane. Impermeable, we mean it can't travel through. So we'll see that word a couple of times. Particularly, we'll see that word regularly. Our third type of molecule we're thinking about in the plasma membrane are proteins. So proteins are made of amino acids. They're linked together with their peptide bonds. Then we did a bunch of folding to give them different shapes, different sizes. Okay. So we're going to see a couple different types because proteins in the body have lots and lots of different jobs. So what we're focusing on here right now are integral membrane proteins. When we say integral, we're thinking like integrated, right? So they're like embedded in the membrane. Right, so you can see some integral membrane proteins that go all the way across, like this big peanut one. You can see some integral membrane proteins that only go partway through, but they're both integrated into the membrane, so they're both integral proteins. Okay. Now, when we have proteins embedded like this, this implies, based on their location, right, that they're going to have amphipathic properties just like our phospholipids did, right? Because if we're looking at this protein, it's got a part that's touching water, and it's got a part that's touching those fatty acid tails in the center of the membrane, right? So it's going to have both hydrophilic and hydrophobic parts. Hydrophilic. Sorry, everybody. I pasted the image on top of that word. So they're amphipathic. They've got regions that have both properties. And this is going to be useful for us, particularly when we start to think about transport for moving molecules across membranes, because we're going to see that the center of a integral protein that goes all the way across and is therefore called a transmembrane protein, so across the membrane, is going to be able to have different properties in the center as well, right? So we know we got a polar part on the outside and on the inside where we're touching water, so our hydrophilic part. We know we have a non-polar hydrophobic part where we're touching the phospholipid fatty acid tails. And then in the center of a channel, or in the case of some other types of protein carrier, it's going to be doing other types of transport. We're going to have a region that interacts well with whatever molecule we're moving. So we're going to see channels are often involved in moving ions, which have charges, right? So we're going to have usually some polarity there in our channels. carrier proteins are going to be attracted to usually kind of specific types of molecules. Might be ions, might be other things. So our transmembrane proteins are the ones we usually think of, the big ones that go all the way across, trans across, opposite. But we also have some proteins that are integral, so are embedded, but are only on one side, right? So here you can see our example of that. So these are going to often act as enzymes or receptors. If you had one on the outside, that's usually where you'd find a receptor. So they'll like interact with the environment either inside or out the cell, but they don't have to go all the way across necessarily, depending on what they're doing. In fact, some other proteins just kind of sit on the membrane. So you can also see peripheral membrane proteins here. So here we see our peripheral membrane. So peripheral membrane proteins are just kind of sitting on top of the membrane, whether that's on the outside of the cell or the inside of the cell. When I say on top, I mean they're outside those polar heads, right? So you have the full phospholipid, and then we have this peripheral membrane protein. So they could be on either side of the membrane, but they're often going to be on the cytosolic side, aka the inside, touching the cytosol, because a lot of them are involved with the cytoskeleton. So the cytoskeleton, basically like the bones of your cell, right, holding kind of the shape of the cell, allowing for the cell to change its shape and then move. And this is the purpose of a fair number of our peripheral membrane proteins. So that takes us to the third type of macromolecule that's involved, right? So we had two types of lipids. We have the phospholipid, we have the cholesterol. We have membrane proteins generally, integral if they're integrated in, peripheral if they're sitting on one side. Transmembrane proteins are a special type of integral membrane protein that goes all the way across. Now we got our carbohydrates. So our carbohydrates are usually going to be just kind of tacked on to another molecule. So they often their function is often in like cells recognizing each other or forming a layer called the glycocalyx, which we'll never talk about again, but does exist in certain types of cells. So here you can see what a carbohydrate looks like on a cell membrane. So this would be a glycoprotein. The glyco is for the carbohydrate part. So glyco like glucose, doesn't have to be glucose specifically, but we can associate those words. So our sugar proteins. And we can also have glycolipids. So here we see an example of one that's attached to one of our phospholipids. So that would be our glycolipid. So we won't talk extensively about these in this class, but you should know that they're there, that the membrane does have some carbohydrates as part of this fluid mosaic model. Okay. So when we think about why we care about membranes, we're thinking about physiology, so we're thinking about what they do. So membranes are going to be important because they're going to let stuff cross, and they're going to stop stuff from crossing, right? So they're going to allow for transport. That's the piece where we move things in and out of the cell. And then they have this selective property. So they're selectively permeable, we're going to say, because we don't want everything to get into our cell. We want to control what comes into a cell. Right? We're maintaining homeostasis. We know it's important for cells to be in a consistent environment. So that's going to include what's inside the cell. We're going to be trying to keep concentrations of specific ions, specific slates constant or within our desired zone. So it's important that membranes can block stuff, and it's important that membranes can allow stuff in a regulated manner. So when we're thinking about membranes in general, we're not going to be drawing out all the pieces, right? We're usually going to just be drawing kind of a circle unless we're thinking about a specific type of cell. So when we draw that circle, what we're actually imagining in our heads is like a sphere, right? So we're thinking about a ball with this kind of crust. We're separating two of our types of fluid, right? So when we talked about fluid compartments, we talked about, I'm going to do intracellular fluid first. So intracellular fluid inside cells. And we have our extracellular fluid outside cells. And I'm doing that in a second, so that we have a reminder that our extracellular fluid comes in two types, right? So usually we're going to be thinking interstitial fluid, so ISF. That's probably what we're drawing. But it might be plasma if we were thinking about a red blood cell. So those are two options for extracellular fluid. And we are rarely going to be specifying, because it's usually just interstitial fluid, right? So ECF, extracellular fluid, is enough for us for right now. So when it comes to your exam, if I ask you what our two types of extracellular fluid are, you want to know. Okay. So this means we're going to be able to change our concentrations of different chemicals, particularly inside the cells, but also outside the cells. Right, so we're going to be able to trade stuff between our extracellular fluid. We might draw something in, or we might take something from our intracellular fluid and expel it out. Right, both of these are possible. We can move stuff across the membranes. Okay. Now, this is important because a cell does not have everything it needs inside that cell from the beginning of time and stay that way. We know that from our daily lives, right? We think about breathing. Why are we breathing? Well, one thing we're doing is breathing in oxygen because our cells need more oxygen. They run out, right? So transport is going to be important for pulling things in. Right? We're going to want oxygen to come in. We're going to see other nutrients we want to pull into cells as well, like glucose, right? We might want to pull in some glucose so we can do aerobic cellular respiration and actually get some ATP to run cellular processes. We're also going to want to be able to get rid of stuff, right? So we're going to have waste products build up inside cells as we're running our daily processes. So in particular, we've already thought about carbon dioxide. That can be a common sort of waste product that we think about. And the other part of breathing is you're breathing out CO2. It's actually really dangerous if you were to let CO2 build up extensively in your body. So it's important that we can get rid of stuff. So this is why transport, this movement, is important. Okay. So when we're thinking transport, we have a membrane that's selectively permeable, so it can select what is permeable to, it can select what is able to cross. We've done that largely by picking what macromolecules are forming the cell membrane, right? So that behavior of the phospholipids is really a constraint for us. But we already know, as we mentioned, aquaporins allowing water to move freely, right? We can change it up based on what proteins we have in our membrane. So when something's permeable to a molecule, that molecule can cross. But we also have these restrictions, this selectivity. So when we think about what can and can't cross at baseline, right? So like when we start out with a cell, we're not specifying any specific extra processes. What's easily gonna get transported across the membrane, we can easily move small stuff and non-polar stuff, right? So that's why we're paying attention to the phospholipids, and in particular, that thick layer of hydrophobic fatty acid tails that are non-polar, right? So anything that acts in a polar way, acts like water, is attracted to water, is likewise going to get repelled by those fatty acids that are trying to escape water. So it's not going to be able to get through our membrane. But if you've got a little non-reactive, non-polar gas that's nice and small, like oxygen, like carbon dioxide, we don't actually have any problem just kind of slipping through the phospholipids. So non-polar molecules are easily transported across the membrane. So we're going to refer to that as diffusion, specifically simple diffusion. We'll talk about the different types of transport in a little bit, but that's our kind of preview. So usually when we're thinking about that, we're thinking about oxygen and carbon dioxide, but fatty acids could do that too. You rip off fatty acids off a triglyceride, they're going to be able to easily cross that phospholipid barrier, that cell membrane as well. Now, anything with a charge is not going to get across the membrane easily on its own. So we're thinking ions, right? So we know we've got cations, we've got anions. Right? So ones in particular we'll see a lot in this course are sodium. It's going to be an important ion for us. Potassium is going to be an important ion for us. In terms of, we'll do one more cation. We'll see calcium. It's going to be pretty important. That's usually 2+. Okay. And on the anion side, usually what we're going to be thinking about are chloride ions with their negative charges. Okay. So these charges mean that ions, you put them in a glass of water, they mix in really well, they dissolve really well because they are attracted to the little charges that build up on water molecules. So we're going to have attraction to the sort of separation of charges that we see in a water molecule. So 2 is polarity. But they're going to have the opposite effect when we're thinking about how they're interacting with the fatty acids on that phospholipid. So they're going to be repelled by our plasma membrane. Now that's going to go for other polar molecules too. So you may not have a full charge, right? You may not be an actual ion. But anytime we see a molecule where you have like an oxygen sticking out, right, or an OH group, right, this is going to lead to some polarity because oxygen atoms kind of hog electrons a little bit. So while we have covalent bonds, those electrons aren't really shared equally. So we have like little mini charges here, right? We wouldn't draw them on because these aren't ions, but we're going to have little mini charges. So they're going to interact well with water, right? Our H2O, right? It's covalent bonds. In water, we can see that oxygen kind of sticking out. So it's got like a little mini charge, a dipole moment here. And then we have our hydrogens. We're gonna have like our opposite little mini charge on them. Plus sign. And positives and negatives attract. So we have opposite charges attracting. So when we say that water is a polar molecule, that's what we're referring to. Because we've got this kind of plus region and this kind of minus region, even though we don't have a full charge because we've got covalent bonds, not ionic ones. And that's leading to these attractions with other molecules. So anything that's ionic, anything that's polar, isn't going to be able to cross our cell membrane without help. So that's our ions, that's glucose. Proteins are too big apart from also having some of these polar regions. Part of why we are thinking about these membranes, ultimately we're going to think about transport and the types, like what can cross with energy, what can cross without energy. But first we're going to have to understand what direction, the favorable direction, like if you have an ion, if it could cross a membrane, what direction would it go? Okay, so we have to understand that before we tack on are we going to use energy to move, for example, our ion or not? So that's where we're going to be talking about gradients a lot when we talk about our types of transport. So when we talk about passive transport, passive meaning not active, right? Like not doing anything that involves energy. It's going to be a spontaneous process, meaning it's going to happen without the addition of energy. And we're often going to be describing passive transport as moving down a gradient. So when you're thinking passive transport, you're thinking gradients, you're literally thinking of like a ball rolling down a hill. So when we talk about gradients, which we'll very shortly, we're going to be describing things moving from high. For right now, we'll use the example of concentrations, high concentrations to low concentrations, right? Which is why we've got this hill visual in mind. So we're going to be moving downhill, we're going to be moving from high to low. Active transport is the opposite, right? So in active transport, you are trying to roll this ball up the hill, right? So when we roll a ball up the hill, we need to use energy, right? So in active transport, you go from, for example, if we're thinking about chemical concentrations, from a region with low concentration to a region with high concentration. We're thinking about rolling the ball up that hill, depending how big the ball is, depending how big the hill is. It's going to require different amounts of work, but if you get a big enough ball, a big enough hill, that's going to be hard. So this is what's going to require energy. Our active transport is going to require an energy source. It's not going to be spontaneous. We're going to have to put in that energy from somewhere. Okay. So now we're going to be thinking about driving forces. Okay. So one piece of this you've probably thought about before in other classes. One piece of this is probably going to be kind of new. So we're going to be thinking about both chemical forces and electrical forces when we think about which way we like down the hill, which way is passive, which way is easy, which way is going to be up the hill for transport, which way is going to require energy, which way is kind of high. So what you probably talked before about are the chemical forces. So when we say something moves from high concentration to low concentration, that's what we're talking about. We're talking about a chemical force. So concentrations are like collections of chemicals. But we're also going to have electrical forces. So like those charges on the ions, right, our positive cations, our negative anions. Those forces attract each other, right? So positive charges are attracted to negative charges are attracted to positive charges. And if you have two charges that are the same, so positive, positive, or negative, negative, they repel each other, right? They separate, they push each other apart. So imagining taking two magnets and trying to push them together, right? And you got the orientation where they snap together and get really hard to pull apart, depending on how many magnets. But we've also got that end where you're trying to push them together and you can feel like an imaginary force field kind of stopping you. So we're going to have those electrical forces also acting on our cells. So we are going to learn how we build up an understanding of the electrochemical force. So electro for this electrical part, chemical for concentrations of chemicals. And we're going to get one big sum of all these forces, electrochemical force. And when we ultimately determine, okay, is moving this ion in this direction passive, or is it active, is it going to require energy or not, we're going to need to know, is the electrochemical force, so this total sum of all the forces acting on this ion, which direction does that go? So if you go in the direction that the electrochemical force is pushing you, then you're going to be moving in a favorable direction, the direction it likes. So that's going to be our downhill movement. That's going to not require energy. But if we have an electrochemical force that's pointing the other way, so whether that's for chemical reasons, right, so our concentrations, or the electrical reasons, those charges or both, if we have these forces adding up in such a way that they're pointing like into the cell, but we're trying to get an ion out of the cell, we're going to have to resist this electrochemical force. So we're going to have to do active transport. We're going to have to find energy and use it somehow. So that's why we're talking about our driving forces before we go through the different types of transport. We're going to start with the chemical driving force, with the understanding that for our ions, we're going to have to also add in our electrical driving force. When we're thinking about a chemical driving force, this is what you hopefully talked about before. we have our molecules, our ions, our solutes, whatever we're paying attention to, going from regions where we have high concentration to regions where we have low concentration. This is basically like, they want to be all spread out, right? They don't want to all be sitting at the same table and then have like 10 empty tables around them, right? So we have movement, kind of unpacking, right? So in this image A, we can see we've got a lot of our molecules packed into the cell, relatively sparse outside the cell, right? So we'd say we have a high concentration inside, right? So high concentration, we got a lower concentration outside. So our chemical driving force, which we're going to refer to as our CDF, chemical driving force, is going to have a favorable direction where we push this molecule out of the cell, right? So when I say the chemical driving force points out, or the CDF points out, this is what I mean, right? What I mean is that we've got more of this molecule inside, we've got less of it outside. So overall, what that molecule wants to do is to move out so everybody's evenly spaced. Okay. So more formally, we would say that what's happening here is that we're reacting to a difference in particle concentration between the inside of the cell and the outside of the cell. We have this force that's pushing our particles from higher to lower concentration. And this is going to be different strength, actually, depending on how big a difference we have in those concentrations, right? So if they're pretty close in concentration, this is going to be a relatively weak force. If they're super different, like when we think about the mitochondria, putting all those hydrogen ions in the intermembrane space right before we make ATP, so this can be really powerful, right? So those molecules are really going to want to move, right? So that's why in oxidative phosphorylation it works so well. We've packed a lot of hydrogen ions, trapped them in this intermembrane space, and then when we let them move, they rush through. So here we have an opposite example, right? So chemical driving force can point out, can point in. Here we have a chemical driving force, or CDF, pointing in. But we're always thinking high concentration, right? Lots of them, to low concentration, not very many. For the electrical driving force, we're going to be thinking specifically about ions. So you don't have to worry about the electrical driving force if you're thinking about like glucose or a fatty acid. We're really thinking about ions. We're going to be thinking about sodium. We're going to be thinking about potassium. We're going to be thinking about chloride. So charged particles, whether those charges are positive or negative, right? So we've got our cations, right? T for that plus. We've got our anions. I'm going to pick out the N for negative, right? Both of them are going to have to respond to this electrical driving force. They attract each other, they repel each other, and we'll pick up here and think about how they're creating something called the membrane potential and then responding to it.